Good morning, everyone. We have studied the cells of the nervous system. There are two types of cells. One is neuron, another is neuroglia. So we have studied about the structure and function and classifications of neuron, but we couldn't get time for discussing about neuroglia. Neuroglia, also termed as glial cells. These are what? These are specialized connective tissue cells found in association with neurons in the nervous system. They are not excitable. Neurons are excitable, but they are not excitable. So they are also unable to conduct electrical impulse. Number of neuroglia cells in yesterday we told this is more than 10 times in number of neuron. They have the ability, neurons have no ability to divide since they are not having centrosome. But these cells can divide, they can increase their number. Neuroglia cells, their function is to support the neurons, support the nervous system. Okay? Neuroglia provides metabolic support and immune protection to neuron. So these are, in short, the functions and what they are actually. So the glial cells, we can divide the glial cells into 6 types. The glial cells present in central nervous system, that is in brain and in spinal cord, they are of 4 types. One is microglia, another is astrocyte, the other one is ependymal cells, and the other one is, last one is oligodendrocytes. So, neuroglia under central nervous system, so you will be getting 4 types of neuroglia. What are these? These are microglia, astrocyte, ependymal cells, and oligodendrocytes. And in peripheral nervous system, you will also get neuroglia. They are of 2 types. One is Schwann cell, and we know the Schwann cells are responsible for myelination of the nerves. Another is satellite cells. Now we will see we'll discuss about the details. The functions of the together, all the neuroglia actually performs these functions. What are these? They create blood-brain barrier. Blood and brain tissue, barrier in between blood and brain tissue. The blood present in, that is the capillaries present in the brain, the inner endothelium lining is much protective. It doesn't allow, very much protective, and it protects the brain tissue from infection by some organisms. Okay, so by that they by creating this blood-brain barrier, they protect the brain cells. Envelope synapses and the surface of the nerve cells. In the synapse, what is axon nerve? This is synaptic nerve, and suppose this is the cell body. This synaptic nerve can make connection with other neurons, either in between cell body and synaptic nerve, or in between dendrite and synaptic nerve. Exodendritic, axosomatic, this sort of synapses can be formed. So suppose this is axosomatic. There is no direct connection. In between there is a cap, discontinuous. So in this cap there is a fluid, extracellular fluid. Okay. This cap is covered by neuroglia, so that this part cannot communicate it, or from outside this is not disturbed. So they protect the synapse. So there another function of neuroglia is protection of synapses by enveloping them. They produce substances that are tropic to the neurons. These neuroglia produce some proteins, and these proteins are important for the neurons, for their development of the neurons, for maintenance of the neurons, for survival of the neurons. So they produce this, neuroglia produce some proteins, and these proteins are essential for the nerves, for the neurons. Why? For their growth, for their maintenance, for their survival. And provide neurons with lactate as an energy source. So we know in the cell, cell wants to derive its energy. So resource is what? Substrate is what? Glucose. Initially the glucose. Now the glucose in the cytoplasm, that is in the neuroplasm, they undergo glycolysis. And the end product of glycolysis is what? Pyruvic acid. If the condition is anaerobic, then this pyruvic acid is converted into lactic acid. So these lactic acids produced by the astrocyte can be provided to the neurons for generation of. Now this lactic acid can be oxidized back into, means lactate can be converted again into pyruvic acid. Now pyruvic acid is converted into acetyl-CoA. This is oxidized in the TCA cycle. So energy can be produced through TCA cycle. Okay. So these cells, glial cells, they can provide lactic acid to the neurons from their own metabolism, from the metabolism of the neuroglia. From the metabolism they can produce lactic acid, and they can provide this lactic acid to this neuron, so that these cells can derive their energy from the lactic acid. That is another function of the neuroglia. And synthesize neurotransmitter precursor for neurons. We know these neurons produce neurosecretions, neurotransmitters like acetylcholine, GABA, adrenaline, noradrenaline, glycine. Glycine is an amino acid, it is also neurotransmitter also. So these neurotransmitters are produced in the endoplasmic reticulum. They are packaged here, forms vesicle. So within the membranous vesicle, within this there are neurotransmitters. Now these neurotransmitters will pass through the exoplasm to the axon nerves. When these axon nerves are stimulated, when action potential arrives here, these lot of synaptic vesicles you will get there. When nerve impulse arrives here, what happens? These vesicles moves towards this membrane. This is the presynaptic membrane. So here they attaches and their membrane diffuses. So now the neurosexual section within this will be liberated in the extracellular fluid in the synaptic cleft. And for them there are receptors here in the postsynaptic membrane. So these acetylcholine or norepinephrine, they will bind with this, and here it will increase the permeability to sodium ion, so that action potential is generated. Today we will be discussing about how action potential is formed. What is action potential? So, for the production of these neurotransmitters, some raw materials are needed. The neuroglia can provide this raw material to the neurons. From these raw materials, the neuron can synthesize their transmitters. So we can see neurotransmitter, these neuroglia, they are performing some very important functions for the neurons, means for the nervous system. These phagocytes, they are also phagocytic in nature. So they provide nutrition, they create myelin sheath, they act as set point of regulation of various neuronal control mechanism and responsible for the fixation of the neurons within the nervous system. So these are the main important function of the neuroglia. So within this tissue, see, within the nerve tissue, see, this is the neurons and they are supported by lot of neuroglia, different types of neuroglia. Now let us learn about astrocytes. Found scattered throughout the CNS central nervous system, present in grey and white matter. In the brain there are grey matter, white matter. In the spinal cord also you will get grey matter, white matter. This is the section of the spinal cord. This is the back side, this is the anterior side. This is the ventral side, this is the anterior side. So there are two roots. Posterior root. All the nerve fibers from the periphery enters in the spinal cord through the posterior roots. And the nerves coming from the brain, they are descending to the spinal cord and they leave from here to this anterior root. So anterior root supplies the nerve to the periphery and posterior root collects information from the periphery and send it to the brain or make the center for reflex actions. So these astrocytes found scattered throughout the central nervous system, means in brain as well as in spinal cord. Present in grey and white matter. This A-shaped area is grey in color. Means the cells, the neurons here are actually non-myelinated. Mostly non-myelinated. And this area, they look white. That's why they are white matter. The neurons present here, they are having myelin sheath. They are myelinated nerves. So, see, this is capillary in the blood, in the central nervous system. How these are powered by the astrocytes. So they are making a barrier. This is called blood-brain barrier to protect the brain tissue from infection. We are getting a lot of infections, no? But these organisms cannot enter into the brain tissues because of this barrier. If this barrier is broken, now they can enter into the central nerve, they can damage the central nerve. In that case, it is very difficult to survive. What are the functions of the astrocytes? Provide energy, nutrition, and metabolic support to the neuron, removes wastes. Metabolic waste they also removed by the astrocyte. Provide structural support to the neuron, promote tight junctions. When in tight junctions in between the epithelial cells, so that by pressure or by some means we cannot separate it out. So this is for protection, for maintaining the epithelium. This is needed. These are creating a force between the two epithelial lining. These are present. This is one epithelial cell, this is one, this is another. So at the side, these connections will be there. Tight junctions will be there. Regulate extracellular brain fluid composition. Brain fluid means cerebrospinal fluid, and the brain tissues surrounding the brain tissue, there are some fluid, and this fluid, what will be there in this fluid, these are decided by this barrier, because all the substances are not permeable to enter into this barrier. All this is done because to protect the tissues of the central nervous system. When the neurons of the brain of the spinal cord are injured, so they are replaced by the fibrocytes. Here also, if you cut your skin, then you will find this is repaired, and this is repairing is done by the fibrocytes. Even our lung tissue, by any injury or by infection, if it is damaged, that area will not be regenerated by the lung tissues, but they will be replaced by fibrous tissue. In central nervous system, if brain cells are damaged, in the spinal cord, if brain cells are damaged, then they will be replaced by the fibrocytes. And these fibrocytes are formed by conversion of the astrocyte into fibrocytes. This process is called gliosis. Ependymal cells. In our brain, there are some hollow space. There are some hollow space. And at the two sides, these are called lateral ventricles. Two lateral ventricles are there. They are interconnected to another ventricle called third ventricle. This third ventricle is again connected to a neurotubular structure to another ventricle called fourth ventricle. Now the fourth ventricle is connected to spinal cord. This is the central canal. So through the central canal. So what is there in these ventricles? Cerebrospinal fluid. Cerebrospinal fluid. See, this is the brain, and these two are lateral ventricles, and these are the third ventricle, and this is the fourth ventricle, and this fourth ventricle is continued to the central canal. If you take a section of this spinal cord, it will look like this, and at the center you will get this central canal. Now these are having cerebrospinal fluid. Now this cerebrospinal fluid are from choroid plexus here. So choroid plexuses are they produce CSF from blood, a specialized fluid for circulating, for providing nutrition to the brain tissues. Okay, the brain tissue will get their excretions here, their metabolites here, they will get oxygen from here. So therefore, these are very important. CSF is important. So now what is the function of these ependymal cells? These ependymal cells actually line the ventricles, third ventricle, lateral ventricle, they line it and they secrete cerebrospinal fluid. So in the choroid plexus, actually the choroid plexus is lined by these cells, ependymal cells, and their function is to produce cerebrospinal fluid. So this is very important. Specialized macrophages are seen. So we know the macrophages are having they are phagocytic in nature. So if anything abnormal, anything cell is dead, they immediately captures it, destroys it. So that is their function. Perform a housekeeping function. Means, as I said, if some cell is dead, so it has to be removed. If a person in the family dies, do we keep the body there? No, we remove it. So these cells, they actually remove these dead cells. These are microglial cells. Then oligodendrocytes. Oligodendrocytes, these are oligodendrocytes. These are present both in gray and white matter. Their function, these cells responsible for myelination of reactions within the central nervous system. In the peripheral nervous system, who is doing myelination? Schwann cell. So in the central nervous system, if it is needed, in the central nerves are very short in length. So here myelination is done by these cells, oligodendrocytes. See, these oligodendrocytes in central nervous system, how they are producing myelin sheath, Now, in the peripheral nervous system, there are two types of neuroglia, glial cells. So, what are these? One is Schwann cell, another is satellite cells. So, the function we have discussed, the Schwann cell wrap around the portion of the axon to form myelin sheath. Again, there is a gap of 1 micron. Then again myelin sheath is formed. Then again gap. In this way, from the axon hillock, it continues up to the synaptic knob. So that gap in between of 1 micron, it is called node of Ranvier. And the satellite cells encapsulate the dorsal and cranial nerve ganglion cells. Dorsal and no picture is not there. Dorsal and cranial nerves. As I said yesterday in our class, the cranial nerves, when originate from the brain, they leave the brain and passes to the organ, goes to the organ. And before reaching to the organ, they end in a ganglia. The ganglia is what? The aggregation of some cell bodies. So these cells will be terminating there by producing synapses with these cell bodies. These nerves from brain to the ganglia is the preganglionic fiber. And from the ganglia, postganglionic fiber, the cell bodies are there. From there, the fresh neuron, fresh action will be formed. This action will terminate into the gland or into the muscle. For example, in gastric gland. So first they will come to the ganglia. From there, through postganglionic nerve fiber, they will end into the glands. So when these are stimulated, so gland will do something through these cranial nerves. So these are the ganglion nerves. So what is the function? These are the ganglia. What is the function of these cells? Encapsulate. They protect these ganglionic cells. They protection, every organ is protected. So this ganglion is also needed to be protected, and this protection is given by the satellite cells. You would also find them in dorsal. I told you here are also some ganglia. This ganglia and that ganglia comes under parasympathetic nervous system, and this ganglia comes under sympathetic nervous system. And the ganglia of sympathetic nervous system lies close to the center. If the spinal cord is the center, the ganglia will lie close to the center, outside the spinal cord, close to. So here preganglionic fibers are short, but postganglionic fibers are long, just opposite to that of the parasympathetic nervous system. Got it? So these ganglia, dorsal root ganglia, they are also protected by satellite cells. So their main function is to protect the ganglionic cells. Either it is of sympathetic nervous system or it is of parasympathetic nervous system. Okay, so that way these cells are very important to us. So you can see these are the cell bodies of the neurons and these are the satellite cells. Lots of satellite cells are there. Within these satellite cell, neurons remains protected. And now in your syllabus it is there, nerve growth factor. So you may expect questions from this. These are actually what? There are various factors that are, these factors are needed for the neuronal development, for their maintenance, for their survival. And what are these factors? We call together them now, we call them, they are of two classes. One is neurotrophins, plus are others. So our nerve growth factor comes under this category, neurotrophins. And there are other factors, these we will not discuss now, you will be learning it later on. We will be confining into the neurotrophins. So neurotrophins are trophic proteins to the neurons. Means these proteins are secreted from other sources and they are collected by the axon terminals of the neurons and they are absorbed. Now these substances are needed for their growth, their maintenance, their protection. That's why we call it neurotrophins are protein and these are trophic to the neurons. Trophic hormones. See, gonadotrophic hormones. Hormones produced in the pituitary, but their action is not there. Their action is where? In the gonads. So, these secretions are trophic to the gonads. Similarly, these proteins are trophic to the neurons. Synthesized, produced by somebody else, now but they are needed by these neurons. Sometimes they also produced, neurons also produce neurotrophins. They promote nerve growth and their survival. These neurotrophins, what is their function? By the name implies nerve growth factor, they promote growth, their maintenance, their survival. Among these neurotrophins, this is most important, and it is the first neurotrophin identified, isolated and studied. There are others, neurotrophins, brain-derived neurotrophins, neurotrophin 3, neurotrophin 4, but you have to, for your knowledge, for this phase, in this phase, your knowledge, you need this knowledge about nerve growth factor. So you may expect questions from this. So nerve growth factor is one kind of neurotrophins. Neurotrophins are what? Neurotrophins are some proteins which are needed for the growth, maintenance and their survival. So nerve growth factors, particularly the nerve growth, what does it do? Nerve growth factor is the first neurotrophin to be identified. It promotes the growth, maintenance, proliferation and survival of sympathetic nerves and some sensory nerves. Nerve growth factor, these are actually neurotrophic peptides, is made up of 2 alpha, 2 beta and 2 gamma chains. These are proteins, so they will be made up of amino acids. So these amino acids remain in a peptide bond. So we will find 2 alpha peptides, 2 beta peptides and 2 gamma peptides. Okay. So now these alpha peptides are having trypsin-like activity. Trypsin is an enzyme that breaks protein into amino acids. It is secreted by, it is also secreted by this trypsin or gen is secreted by pancreatic juice. And their function is to digest peptones into amino acids, into short dipeptides. So here also these neurotrophins, the nerve growth factors, some nerve growth factors which are present in alpha subunits, they act like, suppose we said, they act as, the cell act as sweeping, means dead cell removal. So when they will be phagocytosed, so these are cells, there are proteins, so this protein has to be digested within the cell. So with this trypsin, they digest it. And there are beta subunit, and this beta subunits is like insulin. So we know insulin promote growth. Insulin promote growth also. Besides metabolism, carbohydrate metabolism, it also promote growth. So here also, this nerve growth factor, beta unit, they are having insulin-like activity. So they will provide growth to the neurons. And the last portion, that is gamma unit, these are having the activity like serine proteases. These chymotrypsin, trypsin, pepsin, these are serine proteases. Means serine is a proteases. Means proteases means what? What are proteases? What are proteases? Who can say what are proteases? Proteolytic enzymes. Proteolytic enzymes are proteases. And these proteases needs serine as one of the amino acids, because thus these proteases must have a catalytic site which will break down the amino acids, break down the peptides. This catalytic site must have a serine residue. For their activity, for their catalytic action, they must have a serine residue. Serine is an amino acid. So that's why this gamma subunit is heavy. There are some other peptides. They don't have serine residue in their catalytic site. But for the main enzymes, they must have serine residue. So here also we can see that gamma subunit also having serine residues. Angiols, these are another important function. It prevents apoptosis. We studied apoptosis. So it also death of the cells. So it prevents death of the nerve cells. And where they are produced? They can be produced mainly by the astrocytes in the brain. Other than brain, they can be produced by the glands, particularly salivary glands. They can be produced by the muscles. They can be produced in plasma. We can get it in plasma also. So these are the locations from here where we can get how they are transported. See, produced by various structures, as I said, the nerves will be supplying some structures, and if these structures can produce nerve growth factor, so these structures are producing nerve growth factor. Now this nerve growth factor will be absorbed. They will bind to the receptor at nerve endings. This is the nerve endings, and suppose this is the cell from where NGF is produced, nerve growth factor is produced. So they are secreting it to the exterior. So here are the binding sites for NGF. So NGF will be binding here and they will be endocytosed within the exoplasm. Now they will be passing through exoplasm will be passing to cell body, and there it will produce its action. So in this way, they bind to the receptor at nerve endings, internalized by endocytosis, transported to the retrograde exoplasmic fluid, integrate, retrograde, reverse flow of exoplasm. They will come to the cell bodies, and in the cell body, cell body increases production of protein association with neuronal development, growth and survival. This is the mechanism. How they are transported, how they act. Significance of NGF. Keep a photo of this. Question me, what is, write short notes, or what are the importance, clinical significance of NGF. So already I told, these are the points summarized here. Come. So these are having some clinical value in different disease condition: dementia, depression, schizophrenia, autism, anorexia nervosa, bulimia. So these are some conditions in which NGF can be used as a drug. Okay, so in that way, these are very important. NGF could also be involved in various psychiatric diseases such as this, as I said. Dementia, depression, schizophrenia. Schizophrenia, what is schizophrenia? Dementia, forgetfulness. So there is autism. See, autism, autistic child, have you ever seen? The autistic children, the parents who are having a autistic child, this child is become burden to the parents. Why? When at the early stage, it is not possible to diagnose the baby is having autism. But when it grows, it will never make eye contact. The baby will not make any eye contact. It will not respond to the instructions. No, the baby will not follow any instructions. And sometimes they become very aggressive. The infants become, or the children become very aggressive. So sometimes they injure themselves by knocking their head on the walls. They cannot be educated. You cannot make them educated. They cannot learn anything. So they are very problematic child. So autism, there are some autistic autism society. The parents are making these, all the autism students or children are cared by this society. Okay, so we will not now discuss myelination. How myelination, it got some idea how Schwann cell actually makes myelination. So this is one part, but I can see this is in your syllabus. What is myelinogenesis, how it happens, how the Schwann cell makes myelinogenesis. So this is not included in the, I can't see in the syllabus. Okay. So competency number 3.1 we have finished. Now the competency 2, 3.2, what it says? What it says? What are the different types of nerve fibers? Not neurons. Neurons, nerve fibers. Sir, what is the difference? Who is able to answer? Nerve fibers, neurons. None. Properties of nerve fibers, including strain, duration curve, pronouncing, and reovers. So this is written, but covers so many things. It covers so many things. So I'll discuss, starting to discuss it now. So we'll study organization of the nerve. Axon neuron is having axon. The axon of a nerve is called nerve fiber. Nerve fibers are actually the axon of there may be multiple nerve fiber bundled together. Multiple axons may be bundled together. This bundle having multiple axons are called fasciculi. One bundle is called fasciculus, and if there is number of bundles, then it is called fasciculi. Then again, in a nerve tract, there can be number of fasciculi. Numbers of fasciculi, more than one fasciculi. Again, they will be wrapped by a sheath, membrane. Let's see, what are these membranes? These are what? Fasciculi. And all this together is a nerve bundle. So this is the axon. This is one of the axon, one fiber. And this fiber axon is covered by axolemma. This axolemma is again covered by a membranous structure. This is called endoneurium. What is this called? So you will get just outside the axonema. What you will get? Endoneurium. So one axon having endoneurium, these are myelinated now. So one axon having endoneurium, another axon having endoneurium. So endoneurium, they will be joined so that they are kept in a series, in a line. Okay, now see, lot of this nerve fiber means axons are bundled into a fascicle, into a fasciculus, and this fasciculus is surrounded by a membranous structure. This is perineurium. Here it was the endoneurium covering the each individual fiber. Here the fascicle is covered by another areolar tissue membrane, and this is called perineurium. So perineurium covers the fascicle. Now these fascicles, again, all these fascicles are covered by another membranous structure. These are what? These are epineurium. These are our most outside covering is epineurium. So what is epineurium? We may ask question. It can be given as MCQs, epineurium, endoneurium, perineurium. Perineurium, one correct answer will be correct. So you have to know the difference between these two. Now you think, vagus nerve is coming. These are myelinated nerve. So these are bundled. So in this bundle, lot of fasciculi will be there. Some fasciculi will be going to heart, some fasciculi will be going to upper part of the GI tract, some fasciculi will be going to the middle part of the GI tract, some fasciculi will be ending into the intestine. So in this way these are distributed. Now all these fasciculi, these are having again lots of nerves. When you will be able to stimulate all the nerves in a fasciculi, it will depend on the strength of the stimulus. If stress is much more, then all these nerve fibers will be stimulated at a time and the response will be much more. When the strength of the stimulus will be less, number of muscle fibers stimulate, number of fiber will be stimulated, fiber will be less, and they will stimulate the nerve or the muscles, less number of muscles, response will be less. Now we will classify the main part. Our main part was classification of nerve. There are various ways of classifying nerves. Various ways. How many? You will find. There are 7 to 8 ways of 7. One is depending upon the structure of the nerve fiber. Means if there is myelination in the axon, myelination is a part of the structure, then we will call it myelinated nerve fiber. All these peripheral nerve fibers are myelinated. And the nerve fibers may not have myelination. They are unmyelinated nerve fiber. We will find it in central nervous system. We will find it in gray matter of the spinal cord. So, in dorsal root we may also get. So, there are non-myelinated or unmyelinated nerves. So, depending on the structure, we are dividing them into myelinated and non-myelinated. And depending upon the distribution, where they will be distributed. sensory nerves, we know somatic nervous system. Somatic, peripheral nervous system is autonomic and sensory and somatic. Somatic nervous system is having sensory nerve and motor nerve. So by the sensory nerves, sensory brings sensation from the periphery to the center, and motor nerves makes the response. It sends impulses to the effector organs and produce the action. So these are afferent nerve and these are the efferent nerves. So these are the distribution. Whether distribution means some will be carrying impulses from the periphery to the brain to the center, and some will carry from the center impulse from the center to effector organs. What are the effector organs? Muscles, glands, gastric glands, salivary glands. So they hormonal glands. So these are the effector organs. Depending again with this is the third division. Depending on the what type of neurotransmitter is secreted from the nerve endings. Can you name one of the nerve receptor or neurotransmitter? At the right side of the red sweater. Name one neurosecretor. Neurosecretion. Dopamine. Sit. Mainly they are classified into adrenergic and cholinergic. Adrenergic, the nerve endings, from this synaptic nerve, from this nerve endings, acetylcholine will be liberated. If acetylcholine is liberated from the nerve endings, this nerve will be regarded as cholinergic nerve. These are actually parasympathetic nerves. These are actually parasympathetic. See, hey, contract your bicep. What is happening? My efferent nerve release some acetylcholine and it causes contraction of the muscle fibers. So this nerve is cholinergic and some fibers are adrenergic. At the nerve endings, instead of acetylcholine, adrenaline or noradrenaline, or adrenaline has got another name, epinephrine. Noradrenaline also have another name, norepinephrine. So we can classify the nerves into major group. One is cholinergic nerve, another is adrenergic nerve. See, adrenaline. Suppose now started earthquake. What will be doing? Instantly, what will be doing? Running, no? So, what will happen to your heart? Heartbeat will be increased. Heartbeat will be increased. Who will make this? Sympathetic nervous system. How? From that sympathetic nerve endings, adrenaline will be secreted. Now this adrenaline will cause increased beating of the heart. But on the other hand, we have learned if sympathetic nerve is stimulated, from the nerve ending adrenaline will be secreted, and this adrenaline suppresses the activity of the GI tract. Motility as well as secretions. So their action is opposite here in the GI tract. But in respiration, in heartbeat, there is excitatory. It will cause increase in the function. Again, depending on the diameter of the conduction, this is important. This is important for your exams. Okay, so we depending on the diameter, because the basis of classification is what is the diameter of the bundle, nerve fiber, whether it is myelinated, how if the diameter is much more, then velocity, how fast impulse will be going, it will depend on myelination first, because here conduction will be substitutory method, and the diameter. Higher the diameter, more will be the velocity. Less the diameter, less will be the velocity. Higher the temperature, more will be the velocity. Less the temperature, less will be the velocity. These are the three main factors that determine the velocity of nerve impulses. Let us see. The nerve fibers, type A fibers are also further classified. Let us see what are this classification. The type A fibers are also called alpha fibers. They are also called type 1 fibers. Type A beta, type A is having alpha, beta, gamma, delta variety. Type A, in the previous slide we have seen type A, type B, type C. Now we can see type A is again are of various types, alpha, beta, gamma and delta. So alpha is also called type 1 nerve fibers, beta, A beta is also called type 2 nerve fibers, and D is also called type 3 nerve fibers. Okay, now see the diameter. Higher the diameter, higher will be the velocity. Velocity meter per second speed, and the diameter is unit of diameter is micrometer. If the type 1 A receptor, A fibers, nerve fibers, they are having diameter of 12 to 24 and they are having velocity 70 to 120 meter per second. As the diameter decreases, see velocity decreases. And see the non-myelinated nerve fiber, their velocity, their diameter is less than 1.5 micrometer and their velocity is 0.5 to 2 meter per second only. So this is one knowledge you get. Now means higher the diameter, higher will be the velocity. The myelination, if there is myelination, higher will be the velocity. If there is no myelination, less will be the velocity. Higher the temperature, less the temperature, less will be the velocity. So we have classified the nerves in different. Another type is the Golgi type 1, Golgi type 2, depending on the length. Depending on the length of the nerve. If the length is more, these are type 1. If the length is short, these are type 2 Golgi. Golgi type 1, Golgi type 2, depending on the length of the nerve fiber, we have classified them into two. Golgi type 1, length is more. Golgi type 2, length is less. Third, now the very important part. Very important part. You have to, if you want to, what is the time? Not. I already talked to the anatomy department. They allotted me 15 minutes time more. What happened? You can go. You come out. Number one, how I could allow you. You are having no apron. Number one. Second, you go. Are there anybody who do not want to continue the class? Anyone? Anyone? No. If you can go, I will not mind, you can go easily. Generation of action potential. Our nerves are excitatory in nature. All the body cells are having resting membrane potential. But all nerves cannot produce action potential. All the cells cannot produce action potential. So the cells which are capable of producing action potential, they are called excitable cells. So excitable means the cells are capable of producing action potential. We'll see what is action potential. This is very important. This is very important. Whenever you will be studying heart, this is important. Here, whenever you will be studying GI tract movement, action potential is important. When you will be studying the functions of the nervous system, action potential is important. So everywhere you will get action potential. So sound knowledge regarding action potential you must have. So under resting condition, as I said, every cell is maintaining a resting membrane potential. If there is no resting membrane, the cell is dead. How will you know there is a potential, there is resting membrane potential, there is a membrane potential? So you insert one electrode inside the cell just below the membrane and another outside, and connect it into a galvanometer or cathode ray oscilloscope. You will find, immediately you will find there is a negative deflection, and the point is where? At minus 70 millivolt. In case of nerve cells, it is minus 70 millivolt. So resting membrane potential, what is the resting membrane potential then? Minus 70 millivolt. Now this minus 70 millivolt, this is the under resting condition, there is a potential difference of minus 70. That means inside is more negative than outside. In respect to outside, inside is more negative. How much? Minus 70 millivolt. And this under resting condition, this potential difference across the membrane is called resting membrane potential. And these are created. Why these are created, resting membrane potential? We have discussed it because there are so many factors that created resting, including while we are studying GI tract or general physiology, I have taken this class. How resting membrane potential, mechanism for development of resting membrane potential. See, this is the under resting condition. This is the my goodness, what is happening? Actually, this is the wrong picture. Actually, potassium concentration is higher inside. This is inside, this is outside. Potassium concentration is higher inside, less outside. So when the potassium ion would find scope, it will try to go out because of the diffusion gradient, concentration gradient. Sodium concentration is much more at the outside, less is here. Chloride concentration is high there, and the protein non-diffusible, this is the non-diffusible ion, proteins are bigger in size, so they cannot diffuse through this membrane. So their concentration is inside, it is less. As a result, as a whole, this is not correct. Here it is more, here it is less. Inside potassium is less. So as a result, we can see there is an unequal distribution of ions. Because of this unequal distribution of ions, the charges are also positive and negative charges also, total negative and positive charges are also unequal. As a result of this, inside is more negatively charged in relation to positive charge ions. The negatively charged are more inside. That means the net is what? Net change, there is more negative charge inside. But at the outside there is reverse. Positively charged ions are, sodium ions are much more. So because of that we get a potential difference. And this potential difference as seen in resting condition is called resting membrane potential. We are human beings. We have to respond to various things. You are seeing me, you are hearing what I am telling you, you are tasting foods. Everything depends on these sensations. Something happening in the eye, I am sensing it. I have to do this result. So everything will be requiring action potential. There should be we have to make potential change across the memory. So this potential change is made through some ions. What are these ions? Sodium, potassium, chloride ion, sodium, potassium, chloride ion, and the protein ions mainly. Okay, so these ions have channels. They pass across the membrane. They pass across the membrane through their own channels. All are having their chloride channels, sodium channels, potassium channels. So they are having their respective channels. Now these channels are of different kinds. Some are leaky channels. They don't require any carrier, any energy. So these leaky channels number is less. So small amount of potassium can go out, small amount of calcium chloride can enter. Small amount of calcium can enter from outside to inside. Small amount of potassium can go outside through this leakage. But their movement is less and number of channels are less. So leakage channels are available for all of them. Sodium, potassium, chloride, calcium, all of them. For all of the leakage channels are there. They are also important, though their number is less. And there are some gated channels. Gated means there is channel but there is door. Sometimes there may be one door, there may be two doors. So these channels are having doors, these channels are having gate. The channels having gate are called gated channels. The channels that are now these gates, how this gate will be opened? How this gate will be opened? Suppose this is the only leaky channel for sodium. Sodium tries, concentration is very high at the outside, it will be trying to enter within the cell. Okay, so through leaky channels it is coming slowly. As a result, slowly inside will become less negative. It was minus 70, now it will be minus 68, minus 65, more entry, less negativity. Because this sodium with this positively charged ion will neutralize the negative charge ion, so it will be less negative. When the negative charge inside negativity becomes a particular level, what happens? Some invisible doors became opened at a particular voltage, at a particular potential. So this opening of these additional gates, at a time, lot of sodium ion will be entering. So how these gates are opened? By change of voltage. So if the voltage is changed, this gate will be opened. The other gates will be opened. So that's why it is called voltage gated channel. Some are ligand gated channels. Ligand. Example of ligand? Ligands are hormones or neurotransmitters. So when they will bind, so these cells secrete acetylcholine. This is a ligand. Now there is a binding site for acetylcholine. When it binds with the acetylcholine, this receptor binds with the acetylcholine, immediately what happens? Lot of sodium channel will open. So now sodium ion will enter and make this cell depolarized. So here these sort of channels are called ligand gated channels. Some are mechanical gated. Here I am having receptors. If I stress these receptors, I can feel somebody or one agent has stressed my skin. Immediately impulse goes. So receptors, by stretching it opens some gates. Sodium ion entered. This cell here, the receptors got stimulated. Now the stimulation will go into brain and I will be knowing somebody has touched or stressed. So there are three types of gated channels. What are these? Voltage gated, ligand gated, and mechanically gated channels. And there are some also channels, these are controlled by energy. ATP is required. Sodium potassium pump, these are actually pumps. Calcium pump, sodium potassium pump, we have seen a lot of sodium potassium, and proton pump, as we have seen in hydrochloric acid formation. So there is exchange of hydrogen ion and potassium ion. So these are also pump, depend on ATPs. So through these different type of pumps, actually ions move across. So we now came to know that there are different types of pumps and different types of voltage gated channels, and there are leaky channels. With the help of all these channels, actually electrical changes take place in the neuron, in the muscles, in the GI tract muscles, in the heart muscles. Okay.